Hi everyone. Now we're going to discuss about biodegradation of pesticides. Yes, among the hazardous environmental pollutants, the most prevalent dangerous group of the chemicals are the pesticides. The pesticides adversely affect the natural systems and disturb the biodiversity and ecological stability. So, what is a biodegradation of pesticide? First of all, what is a pesticide? Let's see. So, here the pesticide can be defined as an any substance or mixture of chemical substances intended for preventing or destroying or repelling or mitigating any pests which are damaging our plants and plant products. So, those are being called as pesticides. So, substances meant for destroying or mitigating any pests is going to be called as pesticide. Now, what is meant by biodegradation of pesticide? Degradation of these pesticides by microorganisms and converting them into non-toxic compounds is being called as biodegradation of pesticides. So why we are supposed to have this biodegradation? What happens if these pesticides are going to be present? So there comes the negative effects of these pesticides. The first one is the disturbance in biological equilibrium. So it is going to create a loss of disturbance in our ecosystems and biodiversity, and then they are going to increase the disease susceptibility of the, that means we are being more prone to the diseases because of imbalance in our metabolic activities. Then bioaccumulation or biomagnification, that means they're going to be more accelerating the pesticidal pollution problem. And then development of the tolerance, the pests are developing the tolerance, not we. The pests that we are using the pesticides to kill them, they are going to have the ability to develop the tolerance against these pesticides, which is a very dangerous one. Then the disturbance in reproductive systems, that is both the aquatic life and animals, aquatic life animals and the humans are going to have directly or indirectly of disturbance in their reproductive systems, that is reproduction. And then effects of behavior, then effects on wildlife and other aquatic animals and organisms, and then even on the plants. So these effects of on wildlife, though they are very far away from us, how they are going to means indirectly they are being leached into the water systems, and from the water system they are going to be entered into the wildlife system. So that's how it is going to have very uh, bad effects of these pesticides in our ecosystem or natural system. So obviously there is a need to study about this one. So there comes the concept of our biodegradation, so which are being degraded by the microorganisms. Why we are paying more attention means some of the pesticides are going to have a persistence of more than years together. Like this, for example, take the pesticides of aldrin, chloradin and DDT. These are all going to persist in the environment for more than 15 years. That means today if I am going to add this aldrin to my crop, tomorrow again I will add, isn't it? So that means 15 years from today and from 15 years for tomorrow. So that's how it is going to be of having the long persistence in the soil, which is going to have a very dangerous effect. So these are the few examples of the pesticides and the persistent time in the environment. Then microbial degradation of pesticides. How these microorganisms are involved in degradation means, in view of these adverse effects, we are supposed to read on the natural systems in general and human beings in particular. An approach to biodegrade, biotransfer, and detoxify these pesticides was given a top priority of this microbial degradation, isn't it? So, among the various practices to decrease the loads of pesticides in soil, water, the degradation by microorganisms, that is what we are going to discuss, has given sufficient encouragement for all of us to remove these pesticides from the environment by using this microorganisms. So, for this, Bullock in 1974 suggested four major possibilities for transforming these toxic pesticides into the non-toxic or inactivating those pesticides by microorganism by having some sort of the things, that is the pesticides used as substrate and energy, and the pesticides undergo co-metabolism. What do you mean by co-metabolism? They are not going to derive the energy from it, but they are going to co-metabolize in the form of enzymes or something. Then the entire pesticide molecules or its intermediate can be conjugated with the naturally occurring compounds. Then the pesticide is incorporated and accumulated within the organism. So that means they are going to put that one in the organism itself. So by these four possibilities, the microorganisms may be able to degrade the pesticides. Then what are the important pesticide classes that we are having? Here are the major classes like organochlorine compounds. Then we are having the derivatives of carbamic acid. Then we are having the organophosphorus compounds. So these are the few classes where we come to the preparation of the pesticides. Okay, so these are all the examples of pesticides like lindane, carbaryne, dichlorobos, etc. Okay, then coming to the mode of microbial metabolism of pesticides, how these microbes are able to degrade these pesticides means, here comes the, so the mode of microbial metabolism of pesticides is of two types. One is the enzymatic. So here we are going to have the microbial metabolism, non-enzymatic, not related to our microorganisms. Okay. So coming to the enzymatic mode. So here again it occurs in the three phases by the microorganism. One is a incidental type of metabolism of pesticides which cannot serve as an energy source. That's what I told you, co-metabolism, isn't it? That means they are going to have an enzymes called as hydrolases, oxidases, etc. Here, the insecticides serve as substrates and electron acceptors or donors, but they won't derive the energy. Then catabolism, here they serve as energy source. This is co-metabolism, this is catabolism. Then it is detoxification, serving as a resistant mechanism, so such that it is also going to be have the detoxification. So these are the enzymatic mode of metabolism that we are going to observe in the microorganism. So other than this, how the pesticides are going to be metabolized means by other means is non-enzymatic, like photosynthetic breakdown or contribution via pH change, production of organic and inorganic reactants, then production of cofactors. So these are all not going to be discussed here. So we are majorly discussing only the enzymatic type of microbial metabolism of pesticides. Okay, so this what are the microorganisms that are involved in this microbial metabolism? Let's have a look of the thing. So many microorganisms are belonging to diverse groups are involved in the degradation of these different kinds of pesticide, but majorly here I have given you some examples of bacteria and some examples of fungi, so which on which they are going to have their effect on different pesticides. The same pesticide can be degraded by the different types of microorganisms. For example, check here the Achromobacter, Entidobacter are able to degrade the DDT. In the same manner, the fungi belonging group also Phanerochaeta is also able to degrade the DDT. So that's how the same pesticide can be degraded by taxonomically different microorganisms. So you can have the look of all these bacteria which are degrading the concerned pesticide. In the same manner, here we are having a list of fungi which are degrading this all pesticides. Then coming to the biochemistry of pesticidal degradation. So how it is involved in degrading all these pesticides. The first one is going to be of majorly physiochemical reactions and biological reactions are going to occur. So let's have a look of all those things. The first one is going to be aliphatic carbon chains are degraded by beta oxidation sequence. So all the aliphatic carbons that are present are going to be degraded by the beta oxidation process. And then the resulting C2 fragments, that is whatever the acetyl-CoA that is being formed due to the degradation process, are being further metabolized by TCA cycle metabolism that is occurring in the organisms. Then the substituents of aromatic rings are removed partially or completely. Remember this one. Aromatic ring compounds like benzene, all those things can be removed partially or completely. That depends again on their substitutes that they are present. For example, aromatic ring structures degraded by dihydroxylation and ring cleavage mechanism. But As I told you, aromatic ring containing halogen, nitrosulfonate groups, they are unnatural, so they cause interference in oxygenation and cause recalcitrance. So what do you mean by recalcitrance? Means chemicals that show complete resistance to biodegradation. Okay, so chemicals that show complete resistance to biodegradation are being called as recalcitrants. And if the substitutes on these aromatic rings or the methyl, methoxy, carboxy or carbamyl groups, they can be removed from blocking positions metabolically. It is very easy to remove the substitutes containing these things rather than the substitutes containing as halogens, all these things. Then the saturated rings, that means all having the single bonds, are more refractory than the aromatic analogs. Obviously, it is very easy to break the single bonds rather than the double bonds, and they do not support the growth of any organism. However, they may undergo the co-metabolism. So what is meant by co-metabolism? They are going to be acting as a serving as a substrates, or the electron acceptors are donors. Then coming to the organic molecules to be degraded becomes more complex if it contains the aliphatic, aromatic, acyclic, heterocyclic portions together in combinations. So individually if they are present, it's very easy, but if they are going to have in the combinations, then it is very difficult for the microorganisms even to break it, isn't it? Then coming to the if the molecule are connected by ester, amide or other bonds in the molecule, then the cleavage of such bonds by microbial enzymes is the first step of attack, and then subsequent degradation of moieties occur. So these microorganisms are first of all finding the weak bonds like ester, amides and other bonds, such that they are going to first cleave them and then move on to the other groups like methoxy, like these all things. Okay, and even these also going to have the site and mode of initial attack is determined by molecular structure, enzymatic capabilities of microorganisms, and the environmental conditions also. So by having all these things, the pesticidal degradation is going to occur by the microorganisms. And then coming to some examples of this biochemistry of pesticidal degradation, we are going to discuss about the aromatic degradation, mainly by benzene nucleus containing one forming the catechol compounds, and then we will discuss about the degradation of Hinosan, and we also discuss about the metabolism of degradation of DDT, okay, which are the mostly used pesticides. So we will see the biochemistry of those pesticidal degradation. Okay, so let's have. So here is the benzene nucleus. So you know what is a benzene nucleus is. So having a structure like this of a benzene nucleus, okay, single bond. This is a benzene nucleus. The benzene ring is a foundation of preparing many pesticides, and the addition of substitutes such as chlorine, that is benzyl chlorine, or carbon chains, oxygen, sulfur, nitrogen to the benzene ring can provide the compound with its resulting insecticidal properties. So if this benzene nucleus is going to have the addition of any of the substitute, it is going to have the insecticidal properties. And the relative metabolic inertness of benzene nucleus is due to its resonance structure. So we know it is going to have the resonance structure. It is all related to the chemistry, so we won't go into it now. Now, in the aerobic systems, microbes make use of oxygen to hydroxylate the benzene ring and facilitate its cleavage. Whereas in anaerobic system, that means in the absence of oxygen, resonance caused by unsaturated bond is removed by reduction process. So oxygen is present, it is going to be the oxidation mechanism. If oxygen is absent, it will go with the reduction process. In the early biochemical steps of microbial metabolism, aromatic, chloroaromatic phenols and benzoids are generally converted to catechol. That's what I told you. Or if it is a simple benzene, we'll get it into the catechol. If it is a chlorine containing thing, that is chlorophenol or like that, then it will give rise to the chlorocatechol. And some of the compounds like protocatioic acid, for example benzoic acid, COOH, if it is present to the benzene ring, then we are going to get a degradation compound called as protocatioic acid through enzymatic as well as the non-enzymatic reactions. Then see the example of this bacterial catabolism of benzene, how it is doing. So let's have this one. in the early biochemical steps, so as I told you, as a majority of the pesticides with various substituents on the benzene, the catabolism of benzene attains significant in the biodegradation of pesticides. So majority of the bacteria are the successful catabolizers of benzene. So most of the bacteria that we are being used in the biodegradation process are mainly targeting this benzene ring. Why? Because I told you that benzene ring is the foundation of many types of pesticides. So like look this figure, you can see here is a naphthalene compound. Finally, you are going to get a catechol by degradation, and here is a catechol which is further being cleaved into muconic acid and some sort of other muconic acid, so where again these are being cleaved and giving rise to acetyl CoA compounds like that. So this is how whatever the benzene nucleus is, it is being degraded into the catechol compounds. It may be chlorocatechol or it may be of a protocatioic acid, whatever it may be. Okay, so next coming to the metabolism of DDT. So as I told you, DDT is going to be an important chlorinated compound. Okay, and the full form of DDT is, what is it? 4-chlorophenyl 2,2,2 trichloroethane, and its degradation by Phenerocheta chrysosporium. So it's a fungi. Is well established chlorine substitutes makes DDT more resistant for degradation. And what are the other things that is going to degrade this DDT is, one is Enterobacter that I told you, and the one is the Achromobacter. Okay, this is a fungi Phenerocheta, and make DDT more resistant for degradation. However, the production of two lignolytic enzymes, namely lignin peroxidase and phenol oxidase, are the two main enzymes that are present in these organisms, which are able to degrade this DDT, which is going to be of a persistent to degradation. But with the help of these two enzymes, it will be converted into mono and dioxygenase, convert these things into carbon dioxide. That means there is the release of carbon dioxide by the breakage of all these products, ring products. Okay, so this is the structure of DDT. So where going on the degradation, you are going to get your final product, this DBP, that is a dichlorobenzophenone, is further being cleaved. Okay, these rings are being cleared very easily, and you're having the liberation of the carbon dioxide. So this is all the chemical structures from the DDT, that is trichloroethane, 4-chlorophenyl 2,2,2 dichloro, sorry, trichloroethane. You're going to get the DBP, which is a dichlorobenzophenone. So this compound is a benzophenone, and now this rings are being cleared and liberating the carbon dioxide. So ultimately the carbon dioxide will be liberated into the atmosphere. That's how this DDT is being removed from the environment. And then one more example that we have to discuss is metabolism of Hinosan. So Hinosan is an ethyl diphenyl phosphorodiethioate compound, an organophosphorus fungicide, okay, degraded and converted into non-toxic orthophosphoric acid. That means that Hinosan is going to be converted into the orthophosphoric acid by the mechanism of this particular resin. Again, it's a fungi. Okay, so along with this, recently native all these Pariclaria, Enterobacteria, Achromobacter, that means all wild strains of the microorganisms are being treated as less efficient of pesticidal degradation. So in view of fast accumulating pesticides in the environment, we need the efficient strains of microorganisms that can degrade a wide range of pesticides, that too at faster rate, isn't it? So they have to do it fast because of more accumulation of the fast accumulating pesticide. For the last few decades, efforts have been made to alter the genome of the selected microorganisms like Pseudomonas, all these things, so such that they can degrade the pesticides very efficiently. For that, in 1979, a scientist called American scientist, but he is Indian born, but American scientist, Anand Mohan Chakrabarty, isolated or developed genetically engineered Pseudomonas putida, which is going to be considered as a super bug that degrade pesticides. That means why we call it as a super bug means it is containing so many plasmids. That means one plasmid is going to degrade the octane, hexane, decane compounds, and one plasmid can degrade the xylene and toluene, and one plasmid can degrade the camphor, and one plasmid can degrade the naphthalene. So this is how it is present in different bacteria. But all these plasmids were put together and kept in a one bacteria which is being called as superbug, and that superbug is a genetically engineered microorganism nowadays which is used in the degradation of pesticides. So that's how we finish the biodegradation of pesticides. Okay, I hope you understood well. Thank you.